click for biology, we're looking at the concept of stem cells, their differentiation through gene expression, and their application in therapy. There is a PDF of this presentation linked into the description below if you want to download that and add your own notes to it. So the idea begins with the zygote, the single cell produced through fertilization in sexual reproduction. And the zygote undergoes many rounds of cell division called mitosis. And this results in a structure called a multicellular structure called an embryo. But the embryo is initially, at least, unformed in its shape. The cells of the zygote and of the embryo are what we call totipotent. That is, that they can divide, and in principle they can specialize to become any cell which is typical of that organism. However, as we move forward in development, the embryo undergoes what we call gene expression. Now, what this means is that within some cells, genes will be switched on, which will be able to create structures that we associate with the head, for instance, and then other cells will be switched on. Other cells will switch on genes, and those genes will produce structures that we associate with legs, and then every other cell within here can switch on particular genes that will result in their differentiation or cell specialization to form the tissues and the organs of the fetus. Now, as we move forward from the fetal stage into the adult stage, it has been discovered that within adult organ systems, there are retained what we would call adult stem cells. These are called multipotent. Now, multipotent is different from totipotent in that, for instance, if we have stem cells, adult stem cells of the heart, then at the appropriate time when the heart tissue has been damaged, those stem cells can divide, differentiate, and specialize to become particular heart cells and tissues. What those adult stem cells cannot do is to differentiate into cells which are known for other organ systems. And so the heart stem cells can't produce pancreatic cells or liver cells. In the pancreas will have its own stem cells, and the liver will have its own stem cells, all of which are capable of dividing to produce cells typical of that organ, and that would be done when those organs are growing or because those organs are in a state of repair. The ability of cells to differentiate, the stem cells' ability to differentiate, has led to the development initially of therapies associated with stem cells, stem cell therapies. This is very much in its early stages. In popular press, great claims are made about what stem cell therapies are actually possible. These should be treated with caution. This is a technology which is still at its early stages. There are certain advantages and disadvantages associated with stem cell therapy. Let's start with advantages. The ability of stem cells to produce new specialized cells allows organs and tissues to recover from trauma or damage, which has occurred. And so if we can treat the organ or the damaged area of the body with stem cells, we can replace and repair the damage which has been done. One of the disadvantages associated with this is where we can use embryonic stem cells. These embryonic stem cells are usually derived from IVF treatment, where embryos or early stem cells have been decided not to be used for IVF treatment and are donated for stem cell therapy. However, this is seen as an ethical issue, that these embryos may be destroyed in the process of taking these stem cells in order to treat adults, and so that's a negative side. However, that is obviously overcome, where instead of using the totipotent cells of the zygote or the early embryo, that we actually can use the multipotent cells which are already present in adult organs. So that's one advantage and disadvantage. A second advantage of stem cell therapy is that they are particularly good at managing difficult diseases. Sometimes the body's capacity to repair itself is not as potent as one might wish, and if we can introduce these stem cells, we can accelerate the growth and repair of badly damaged tissues, and so that's an advantage. One of the negative sides associated with this is that we don't yet technologically have the absolute control over that cell division. So one cell is dividing into two, and how do we ensure that this cell division doesn't actually become uncontrolled? Because such uncontrolled division is what we call a tumor, which is the basis of cancers. And so that's an important part of stem cell therapy, which has to be overcome and regulated. These are early technologies which are only now just starting to see their applications. Okay, so here's a few questions to check your understanding of this. Only take about 3 minutes to cover these questions. Pause the video, then check your answers and description, and then when you've done that, you can move on to my final screen here. Chance to subscribe to the channel, chance to catch up on the playlist, and if you have the time, why not give this video a thumbs up.